what I would like you to do is open your interactive notebook to page 13. When we talked about meiosis versus mitosis, okay? Meiosis versus mitosis. We're just going to review this a little bit, okay? Who can tell me, okay? Zoe, who can tell me one difference between meiosis and mitosis? Okay, and it underwent, we called that, it had two divisions, right? Meiosis had two divisions, mitosis only has one. So if we look up here on this board, this side is mitosis. You can see we go through, here's prophase, metaphase, anaphase, and it's not including, this does not have every single stage, but if you look at this, when we go through everything, we come through and it has divided once. Now let's look over here at meiosis. Okay, here we got one cell, here we divide it into two, and then during meiosis 2 down here, it divides into 4. So it does indeed have two different divisions. Okay, Linda, what's something different? Give me something else that is different between meiosis and mitosis. Okay, well, they both duplicate the chromosomes. Okay? They both duplicate the chromosomes, but what's something that is different between the two? Okay? We just said that they have two divisions. What else? I can't hear you. Okay? All right. She said, and you couldn't hear it because she's got a soft voice, but what Linda said is that the chromosome numbers were half. So if we're using the terms that I taught you, we're talking about what? Haploid and diploid. So if you look right here, diploid. These are diploid. How do you know those are diploid? It says what? 2N. Now look at these over here. These are haploid, right? How do we know those are haploid? It just says N. Okay? So in mitosis, you are having diploid cells being formed. In meiosis, we're taking it down to the haploid. Okay? Olivia, give me something else that we haven't discussed yet. Mitosis makes body cells. Meiosis makes, what do we call that word? Begins with a G. Gametes. Meiosis makes gametes. Mitosis makes body cells. Excellent. Okay. Anything else? Anything else, Jonathan? Okay. In meiosis, we have some stuff happening like crossing over. Okay? Crossing over. And we know crossing over occurs during what phase? Prophase 1. Okay? Has crossing over. Okay, we're going to talk about something today that we hit on briefly. It's called independent assortment. Okay? Independent assortment. That's something a little bit different too. Okay? Now, the final thing I want to hit on, a difference, okay, is look at these 4 cells right here. Are any of those 4 cells identical to each other? They're not, are they? Now let's look over here. What about these cells? Are those cells identical to each other? Yes, that's another key point. Meiosis has cells that are not identical. The daughter cells are all different. In mitosis, we have daughter cells that are identical. Okay? Daughter cells that are identical. So those are just some ideas, just some review of differences between mitosis and meiosis. Look right here. Here's metaphase. Remember, we talked about the lining up on metaphase, how we have the homologous chromosomes here, and we have the sister chromatid here. A little bit different, aren't they? How they lined up. All right. Now, we are going to go on today and talk a little bit about genetic variation. Okay, genetic variation. How many people in here have a brother or a sister? Okay. Now, okay, let's talk about me, for instance. I have a brother, I have a sister. All right. My brother and my sister have the same parents as I do. Are we the same? Obviously not. My sister and I obviously are not, because she's a female, I'm a male. Okay, so obviously there's some variation there. Do we look the same? Okay, you probably have never seen my brother or my sister, but we don't look the same. Why? The parents are the same. Each are given 23, right? Each are given 23. How's come we don't look the same? Well, it's because of genetic variation. Okay? And we're going to talk a little bit about genetic variation. Let me move my things down out of the way more. I wanted to make sure that we could see the that last slide, so let me move those out of the way. And let's talk a little bit about genetic variation. Meiosis creates this genetic variation. Okay? Independent assortment. We're going to talk about three things right here that create genetic variation. Genetic, what we call diversity. Okay? So one of our objectives, obviously, is to know these three things that create genetic diversity, genetic variation. Okay? One of which is independent assortment. Now, we're just going to list them all right now to start with, and then we're going to talk about each one of them. Okay? So the first one is independent assortment. The second one we've already talked about, but I'm going to show you it, and you're going to draw a picture. That is crossing over. That is crossing over. And the final one that we're going to talk about is random fertilization. Okay? So these three things create genetic diversity, genetic variation. Independent assortment, crossing over, and random fertilization. Okay? Let's first start off and look at independent assortment. Okay? Independent assortment. Now, I'm looking at this right here. The big things about independent assortment, and you have this in your notebook. I left that picture there. What I want you to do is I want you to color your chromosomes red or blue like I have here. Okay? So on that picture, I want you to color these red or blue like I have, and then we're going to talk about it a little bit. Okay? So your diagram should be very, very similar to mine, right? Right? Okay. So go ahead and color those, and we're going to talk about this. Now, we look at this, and let's talk about what independent assortment is. Now, we're dealing with two different chromosomes right here. Okay, a big one and a smaller one. See how in this one right here, both red ones are on the right side and both blue ones are on the left side. Do those chromosomes have to line up like that? Well, look over here. Did they line up the same way there? No. And when we go down to this step, see how it changes based on how they lined up on this step. Can you see how they're different? Okay? Now, you might not think that that's a very big deal, this independent assortment, how they line up when they get to that metaphase 1, okay? Whether they're on the right side or the left side. You might not think that's a big deal just looking at this, but we're only looking at 2 of them, right? What if we had 23 different ones lined up? How many different ways could those line up on that metaphase equator? A bunch, right? A bunch of them. If we're talking about 23 of them, think about how many different possibilities there would be. There would be a bunch. Okay? So, if we look at this, okay, let's talk about that here. Okay? We have human beings. We have 23 chromosomes, don't we? So, if those 23 chromosomes lined up in different ways, okay, 2 to the 23rd, how we figure this out is we take 2 to the 23rd power. When I take 2 to the 23rd power, let me grab my calculator out to give you an exact number. 2 to the 23rd, that value is, okay, 8,388,608. 8,388,608 different possibilities that they could all line up. That's a lot of different possibilities, right? Okay, right? So, think about the variation, okay? When my mom or my dad, okay, when they were undergoing meiosis, the egg cell and the sperm cell have 8 million different possibilities. Can you see why my brother and I don't look alike? Why we're not identical? Why there's variation? Okay? Do you think if you look at your brother, if you look at my brother, that you could see similarities between him and I? Okay? Can you see similarities between your siblings and yourself? You can't? Huh? Right, but some of them you can, right? Some of them you can. So, those different combinations, 8 million. Here is crossing over, okay? We talked about crossing over. It happens in prophase 1. But I want you to get this drawn into your notebook. This is a bigger version. So draw this exact picture in your interactive notebook, folks, and color it exactly that way. So we have a bigger example of this crossing over. All right, when we're talking about the crossing over, here are the homologous chromosomes that are aligned right beside each other. Okay, they're in proximity of each other. Can you see how they got crossed right here? And then when they get crossed, they could possibly switch, switch the genetic information. Thus, we would have what we call recombinant chromatids. Okay? Recombinant because it's a variation between the two. Now, could they cross over at the top also? Yeah. Okay? They could. They could also cross over up here at the top here and switch. So, the crossing over creates some genetic variations. The final thing we're going to talk about here about the genetic variations are random fertilization. We talked about 8 million, over 8 million combinations from mom, over 8 million combinations from dad. So, if we take that 8 million and we take 8 million and multiply those together, we're going to get 64 trillion different combinations that could occur for a diploid zygote. So when the sperm and the egg come together to form the zygote, that is 64 trillion combinations. Now, what do you think the chances are that of those 64 trillion combinations that any two would be the same? What do you think the probability of that is? Not very high, is it? Okay, not very high. All right.